So chapter 20 is largely discussing molecular technology. Over the years, decades, we have come up with a lot of different research technologies that have helped us to figure out what's going on with DNA and to take it a step further into manipulating it. So we're going to go through some of those techniques very briefly in this video, but what I really want you to think about is what natural processes have been manipulated or exploited for us to make these technologies. And I also want you to consider what we can do with them and what are the pros and cons of doing it. Okay, so those are the things that I want you to think about as we go through this chapter. There are subsequent videos that each go through the processes of each one if you want more detail, but I still want you to think about those pros and those cons and what processes are being exploited and the uses of these things. So, as with many things in this course, you might want to consider making a chart. So, the first one is gene cloning, right? And it is, as the name sounds, cloning genes, and genes are those specific pieces of DNA that code for a protein or give rise to a trait, right? So, we can then take and clone them. Well, how is that done? So, gene cloning is necessary to make more copies when we need to do things like sequencing or editing or making probes. And there are two techniques we can try, so either using a vector or a PCR. When you use a vector, you have to be able to clone that gene and then you stick it in that vector that then is put inside of a bacteria in this instance. So that vector is a plasmid, and remember that those plasmids are extracellular pieces of DNA, so it's outside of extrachromosomal, outside of the chromosome, and it gives the bacteria different properties. So here you have that gene of interest, and you can also put it with a selectable marker, so that when you grow it, you know in this case if it's white or blue, if it has it or not. But you then use that bacteria to make more copies of that gene clone. Then you can take them out. And one of the main things that these gene clones are used for are making libraries. So you can make either a genomic library that contains DNA or a cDNA library. And why is this important? Well, it can have all the information from an organism. It works better when it's a smaller organism, but nonetheless it can be very useful. And so here is a chart that just shows you the difference between DNA and cDNA. Now, cDNA is synthesized with reverse transcriptase, so you're going from the mRNA to the cDNA, okay? So because it's mRNA and not DNA, this is now a product of gene expression. And so you can't look at the same things in a genomic library as a cDNA library. So the genomic library is looking at the DNA sequence and all things that it contains, like those promoter and terminators, whereas the cDNA library is expression. So now you're looking at the mRNA that's made and alternative splicing and things of that nature. The next way to do gene cloning is PCR. And so, like the picture shows, you're using a machine here. So with gene cloning, we're using a living organism. With PCR, we're using a machine called a thermocycler to do it. And roughly what happens is you have to know your DNA of sequence, right, your gene. And you want to make more copies of that. Well, you use the system where you have to have a primer. So you design these primers that flank the sequence of interest. Then you put those things in with some single nucleotides as well as a polymerase, which works at a high temperature. And then you set the machine to go, it's going to denature it. So this is like helicase ripping it apart. The primer is going to anneal to it, and then polymerase is going to extend it, and then cycle after cycle with doubling of this product, okay, and however many times you need to do it. And this is all done in a machine. This is polymerase chain reaction known as PCR. There are alternatives to it. So there's reverse transcriptase, where you have RNA of interest, you make that cDNA, and then you make multiple copies. Then there's also real time, where you are going into the cell. I'm sorry, let me back up again to say the end result of polymerase is usually a gel. Well, with real time, you have this signal quencher and these signals, these reporters on there, that's just churning it out. Every time it sees it, it's taking a snapshot. So essentially what you are recording here is a line and how much it makes, okay? So I'm sorry, that's probably not the best explanation, but go to the video for a more in-depth analysis. But the thing to know here is that as it's cycling in real time, as it's cycling, you're getting a readout as opposed to an end product with regular PCR. Then there's reproductive cloning, right? And when you think about cloning, people automatically start to think, no, this is bad because you're making copies of people, right? That's where we want to go. But there are different types of cloning. So we talked about gene cloning. That's cloning. You are making more copies of cloning. Then there's reproductive cloning, and this is the one that scares people because you're essentially making an organism. Then there's therapeutic cloning, which is probably the wave of the future because this is allowing you to make organs, right? So to be able to take adult stem cells from people and then grow an organ in a lab that you can implant in them, as opposed to having millions of people on the transplant list who normally don't survive long enough to get one, right? And so, well, our focus right now is on this reproductive cloning. So reproductive cloning really came to light and to the public's awareness with Dolly the sheep. So there was this technique called somatic cell transfer. And what happened is you take an adult donor and you take out a cell. And then you take another unfertilized cell and you're going to remove that nucleus. So it's enucleated. And you put that adult's nucleus inside of that unfertilized egg. You then, remember that that donor sheep is diploid. The unfertilized egg will be a haploid. So you're removing the haploid nucleus and you're putting in the diploid nucleus. And now it has everything it needs. So you need that cell to then start to form the embryo. Well, you take that and you put it into another sheep or organism that's a female and allowed to be pregnant. So you're essentially impregnating this female. And then she's just going to go normal until the baby is born. Okay, but you're using an adult cell that you take the nucleus out and you put it into an unfertilized egg, allow it to start mitosis, and you put it back in the sheep. And that's her pregnancy, that's her embryo. Well, with Dolly, this is Dolly here. Okay, so she was the first living organism that we know of to be made using this process of somatic cell transfer. And she lived to be, I think, 6 or 7 years old, and she had some issues that they are not saying is related to cloning or whatever. But this was the beginning of an era, okay? So now we are at a point where people are getting their pets cloned, okay? So they had this pet for 10, 15 years, and like animals and people do, it reached old age, it no longer survived. Well, this cat here is called Carbon Copy, okay, and it's the cat that they made that's a clone. So people can, if you have money, you can get your pet cloned. And then, though, the biggest thing is, will we make it to clone people? As of now, no one is saying they have successfully been able to do this. But I want you to keep in mind that monozygotic twins are a natural form of cloning, right? So it happens naturally every once in a while. It can happen unnaturally in the lab, but twinning, identical twins, is cloning in action in nature. But will we ever get to clone people, right? And that's probably $1 million question, and there are a lot of people for and against it for various reasons. And similarly are stem cells. So you hear stem cells and you automatically go wherever it is that you go when you hear these things, but all stem cells are not the same. Right? So there's totipotent and pluripotent ones that can become anything. These are usually fertilized eggs, right? Because they give rise to every single tissue in the body. Then embryonic stem cells and embryonic germ cells from the gonads are pluripotent, where they can become a lot of things, but not everything like totipotent. Then there's multipotent, which can become several different things, and unipotent, which usually become one thing. And so with unipotent, a lot of tissues in the body and like muscle cells have stem cells. So resident muscle cells that if something happens to the muscle, they can turn on and start to make more muscle, but they're only making muscle. Now, a good example of multipotent is going to be like this myeloid progenitor, which gives rise to all of these different blood cells and immune cells. So that gives rise to a lot of different things, whereas uni is one thing, toti is everything, and pluri is many things, right? So when you think of stem cells, all of these are possible. Now, the problem with stem cells is that most people only think about embryonic stem cells. So no, stem cells are bad because this embryo doesn't get to survive. Well, that leaves a lot to people to decide if this is right or not, if it's against religion or whatever it might be. But those come from here. But there are so many uses of it. So is it good? Is it bad? Should we do it or not? It's up to whoever to decide. We have DNA sequencing, and with this is you are really just trying to figure out the order of the letters that exist. This came back in the 70s. It made all the headlines around 2001 because we were able to essentially sequence the first human genome. And there's the Sanger method that many people use, but this is not the only one that exists. And even now, 20 years later, there are easier and faster ways. And the goal is to get to like the $1,000 genome where everyone can be sequenced. But now this too raises questions of what do you do with that information? There's gene editing, so going in and changing things. So we know that DNA gives rise to mRNA, to the polypeptide. And we also know that natural mutations occur, in this case for sickle cell. Single nucleotide change in the DNA changes the mRNA, ultimately changes the sequence of the proteins. It changes the end result. In this case, the shape of those red blood cells and their dynamics. Well, what happens if we can go in and put in mutations where we want them, right? So this is site-directed mutagenesis, and this allows us to then put in the mutation we want to see the effect that it has on a gene, on a protein, or on the organism. There's also a way to do it with CRISPR-Cas9 technology. So this started in bacteria, but now we do it to edit genes, and it relies on DNA repair mechanisms, and depending on the way it goes, it can insert a deletion, or it can gene edit. And this is what people are looking for to be able to correct those diseases like sickle cell, those monogenic mutations. Then we turn our attention to blotting methods. So a lot of stuff that we talked about has been focused on DNA. Well, how do we figure out what's going on with that DNA or those gene products? So if you go back again to the central dogma, we are here, we're focused heavily on the DNA, but how do we then figure out what's going on at the mRNA or the protein level? So we can blot. And so with blotting, there are several different ones. The first one that was discovered was the southern blot based on someone's last name, and that detected DNA. From there, we have the western for protein, the northern for RNA, and even an eastern that does things, though I have never seen an eastern blot done. They largely rely on the same thing. You have to be able to isolate whichever one you're going to do. So DNA, RNA, or protein, you're going to either purify or denature it, run it on a gel with this gel electrophoresis to separate the bands according to size, transfer it from a gel to a membrane, and then blot with that membrane. So either with cDNA, so sequences that are complementary to what you have on your blot, or an antibody and proteins, okay? And then there's some other stuff that's involved, especially if it's proteins. So take a look at that video if you're interested in those specific steps. And then the last one that we're going to talk about in this chapter is allowing us to look at binding. So with DNA and RNA are binding to things. So this is Amenza electrophoretic mobility. So this allows you to detect proteins that are bound to RNA, or transcription factors or proteins that are bound to DNA. And so this is used in gel electrophoresis, but you're not going to denature here because you need that sequence intact. And you're going to run it on a gel, and you're looking for that shift. So what you're looking for is here, this is a DNA that's not bound to a protein. It's smaller, it's going to run further in the gel. Whereas a DNA that's bound to a protein is heavier, and it's not going to be able to travel as fast or as much in the gel. And so you're looking for that shift in mobility to see if it is bound to something. Then you can take this out and run further tests and analysis. Okay, so that's all of the ones that we're going to talk about today. But that begs the question again. So as I was talking, I talked about what some of those processes was. So which process is it exploiting? What are the uses of it? So these are things you can look up in the book and on the internet. And what are some pros and cons? You can look these up, but you can also think about them, right? How would you feel if a scientist was able to clone? Should they do it? Or should they not do it? And why? So that is really what I want you to focus on here, as opposed to each process, because I know that that's daunting, okay? So any questions or comments, or if there's something that you really want to talk about, like let's have a discussion on this during class time.